Okay, can you hear me, everyone?
Yes.
Okay. Now I'm going to revise quickly what we took last time and then continue from there. Last time, we spent most of the session talking about the different experiments, and this was one of the experiments that we talked about. We said here that we will study the effect of light intensity on the rate of photosynthesis. So what did we do? We got a lamp and we placed this lamp at a certain distance from the plant in the beaker. We got a water plant, a pondweed, and we put it in water and we exposed it to light and we measured the distance. Okay. And then we measured the number of bubbles produced or the volume of gas produced, like in this experiment. And we recorded the volume of gas produced over a constant time frame. And then we changed the distance again, and we repeated the same steps. And we talked about all the parts of this experiment. We said that we will create a table with the independent variable on the left side, like we always do. And here, because we were changing the distance, so the distance will be on the left side in centimeters, we will include the units. And on the right side will be the volume of gas produced. Okay, and this is how we carried out our experiment. We said that this experiment can have one source of error, and to avoid that, we placed here a water beaker. What was this source of error? This lamp produces light, and also it produces what?
Heat energy.
Heat energy. Okay. When we created that same experiment, we used that same apparatus, but we can sometimes change the temperature. We can change here, like, for example, we're using here a heater to change the temperature of the water, or we can use different water baths, or we can use different concentrations of carbon dioxide. Now, why are we doing all that? Because we want to prove a certain hypothesis, which is the raw materials needed in photosynthesis. We all agree that the photosynthesis needs carbon dioxide and water in the presence of light and in the presence of chlorophyll to give us glucose C6H12O6 plus oxygen. This is photosynthesis. This is not balanced. In order to balance it, we will apply coefficient of 6 before the CO2, 6 before the water. I will leave the glucose alone, and a coefficient of 6 before the oxygen. And for your information, the opposite of this reaction is respiration. I want to focus on that. Let me open a screen. So our whole lesson is about photosynthesis and the requirements for photosynthesis. So the first thing you will do when you study this lesson is you will study the balanced chemical equation for photosynthesis. Carbon dioxide and water will give me glucose C6H12O6 plus oxygen. I don't think anyone objects with this equation. I'm going to balance it. I will put 6 before the CO2, 6 before all of them except glucose. I will leave the glucose alone. And of course, photosynthesis needs. Now it's gone. I'll do it again. Oh, it's there. Now it needs, we have to have light for photosynthesis. It is essential. This is not nice. 1 minute. Okay. And we need chlorophyll. Okay, because light is going to combine the carbon dioxide and water and give me photosynthesis. Now, photosynthesis has to happen in the presence of light. And this is very, very important because they will give me a lot of experiments and these experiments will be confusing. The moment you see how do you prevent confusion when you have those experiments, let me turn the screen, give me a second. The moment you see light in the experiment, then you will understand that photosynthesis is taking place. If there is no light and there is darkness, then the plant will be doing which process? Photosynthesis or respiration?
Respiration.
Respiration. And the equation for respiration is exactly the opposite of the photosynthesis equation. So respiration will be the opposite. It is glucose C6H12O6 plus oxygen to give me carbon dioxide and water. And again, this is how we balance it because respiration will break down the glucose to give us energy. Okay, I hope this is clear. Okay, going back to my PowerPoint, this is the experiment that we revised. Now let's move on from there so we don't waste time. Now we will move on to the supplement material. And the first thing that you need to know on the supplement material is the definition or the term limiting factor. Limiting factor can be used with plants or it can be used with any other life process. What is the definition of a limiting factor? A limiting factor is defined as something present in the environment in short supply that restricts life processes. So what is a limiting factor? A limiting factor is a factor or anything that will stop a certain process from proceeding. It will stop photosynthesis, it will stop respiration, etc., etc., etc. There are 3 main factors that will limit the rate of photosynthesis. We already know them. We know that photosynthesis is carbon dioxide plus water in the presence of light and chlorophyll to give me glucose and oxygen. So any of those factors, if they are missing, they can be considered limiting. Carbon dioxide, if it's not there, it will be a limiting factor. It will prevent photosynthesis. Light, if it is not there, it will prevent photosynthesis. Chlorophyll, if it is not there, it will prevent photosynthesis. Water, if it's not there, it's going to prevent photosynthesis. It will actually kill the plant. And all the experiments that we are doing, we want to prove that all these raw materials are needed for photosynthesis. We will either remove or play around with the carbon dioxide or the light intensity or the presence or the absence of chlorophyll, etc., etc. Now, the limiting factor. When do you use it? Here, you will use it and you will mention it whenever they will give you graphs. Let's look at this graph. This graph is studying the effect of temperature on the x-axis on the rate of photosynthesis. How did I know? Because I will look at my x-axis and at my y-axis. I am going to study the effect of temperature on the rate of photosynthesis, going back to our experiment, because you will have experiment like this that will take one whole question on paper 6 exam. It will be half the exam. They will give you this experiment and they will tell you that they are changing the temperature of the water. Let's say I'm going to use the other experiment, so we follow exactly the graph. I will change the temperature of the water, and every time I will change the temperature, I will count the number of bubbles. So the table will look like this. What is my independent variable? What am I changing?
The distance of the line.
No, now we're talking about the temperature. If I'm changing the temperature, then what will be my independent variable?
The temperature.
The temperature. I'll write temperature here in degrees Celsius. And every time the temperature changes, I'm going to count the number of bubbles. over a certain period of time. So what will be my results, the dependent variable? If I count the number of bubbles over time, this will give me the what?
volume of
Anything
gas,
that is divided over time, number of bubbles over time. This will give me the
Rate.
rate. Excellent. So the rate will be my dependent variable. Then I'm going to make my graph, and then they will ask you to make I will make my table, and then they will ask you to make a graph. So I want you to understand the graph. In this graph, temperature is on the x-axis, so this is what we are changing. This is my independent variable. And the rate of photosynthesis is plotted on my y-axis. These are my results. And how do I calculate the rate? I will divide the number of bubbles. Always rate is dividing a certain measurement over time. I hope you understand the graph. I will plot my graph, and my graph will look like this. Now, of course, they will ask you to describe and explain and divide the graph, etc., etc., etc. This graph is very easy because we already studied a very similar graph when we studied enzymes. Okay, now let's divide the graph into A and B and C. What will we say at A? How do we describe and explain A? At A, what is happening?
Rate of photosynthesis is increasing at a constant rate.
Okay, you are correct, but I want you to start from the value that is present on the x-axis. As the temperature is increasing, what is happening to the rate of photosynthesis?
Increasing.
Increasing, yes. And then, because this is the describe part, what do we always do to get the 2 marks?
Mention data from the graph.
Yes, you are correct. You are correct. Put up your voice. Be sure of yourself. If I am describing A, describe means I'm going to relate whatever is on the x-axis to whatever is on the y-axis. So A, I want to describe it. It's out of 2 marks. I will take the 2 marks. I will have to say that as the temperature is increasing, the rate of photosynthesis is increasing. This is 1 mark. The second mark, I will give numbers from the graph. For example, when the temperature was 5, the rate was also 5. When the temperature was 10, the rate was 15. I have to give numbers from the graph so that they give me the mark. Okay, very good. Now, this is the describe part. Now, can you explain why is the rate of photosynthesis increasing as the temperature is increasing? Why? We took it before. Photosynthesis is a live process. It is controlled by what? What did you say, my dear?
Particles gain energy.
Yes, you are correct. Which particles? I want to be specific. Which particles gain
Kinetic
energy? Yes, you are correct.
Enzymes and substrates.
Yes, the enzymes and the substrates, the molecules. We said we have carbon dioxide and water in the presence of light and chlorophyll. It will give me glucose, it will give me oxygen. These particles that react together, these particles, the reactants, they gain energy. They collide faster. There is an increase in the chances of successful collisions. And of course, any chemical reaction is controlled by enzymes. So all these particles, all these molecules, when they gain energy, they will gain, when they gain temperature, they gain kinetic energy and they move faster and they collide, and there's a higher chance for successful collisions, more enzyme substrate complex, faster reaction, etc., etc. Give them all the keywords so that they're happy. So here is the explain part. So the describe part, if this question is out of 4 marks, I want the whole 4 marks. Describe, I will first go to the x-axis. Please organize your answer because you lose marks because you missed some information. Describe, I will go to the x-axis. I will find that the temperature is plotted on the x-axis. Then I'm going to start with whatever is plotted on the x-axis. As the temperature is increasing, the rate of photosynthesis is increasing. This is 1 mark. The second mark, I have to give values from the graph, give numbers. Okay, now go to the explain. Why? Explain means why. Why the rate of photosynthesis is increasing as temperature is increasing? The same answer that we took in enzymes, because temperature affects the enzymes. Because as the temperature is increasing, the molecules, like your friend said, gain kinetic energy. We always have to mention kinetic energy with temperature. When the molecules gain kinetic energy, they will move faster. When they move faster, there is a higher chance for successful collisions. When they collide, they will form enzyme substrate complex. When you give them all the keywords, they will be very happy and you will definitely reach the full mark. Okay? Now, what is B? One word. What happens at B? What do you see at point B?
Optimum temperature.
Very good, optimum temperature. This is the optimum temperature. If you want to elaborate a little bit, optimum temperature means the maximum reaction rate. Okay, you don't need to write more than that. Now, what is happening at C?
Enzymes are denaturing.
Very good. This is the explain part. In the describe part, you will say that as the temperature is increasing, what is happening to the rate of the reaction?
Decreasing.
It is decreasing. This is the describe part. Explain, because just like you said, because enzymes are denatured. When they denature, please elaborate so that you take the whole mark. When they denature means what? What do I say when I ever mention denature?
There's change in the shape of the substrate.
Okay, you all spoke at the same time, but you are all correct. When I mention the word denature, then I have to say that the active site is no longer complementary or it does not longer fit with the substrate and no enzyme substrate will happen. Okay, perfect. Now, why did we mention this graph again? Because I want to know from this graph, where is my limiting factor? Or when is the limiting factor here slowing down or preventing photosynthesis? At A or at B or at C?
C.
I will tell you because I do not expect you to know it. At A, look here. When the temperature is increasing, the rate of photosynthesis is increasing. So at A, the rate of photosynthesis is increasing. It still did not reach the optimum temperature. It will reach the optimum rate here at a particular temperature. So at A, what was the limiting factor? What was stopping photosynthesis from reaching its maximum rate at A?
Temperature.
The low temperature.
Exactly. So this is really easy when you understand it. At A, the limiting factor was temperature. Why? Because you can see from the graph that as the temperature is increasing, the rate of photosynthesis is increasing. So the temperature was limiting. The temperature was low. That's why photosynthesis rate did not reach its maximum yet. Photosynthesis rate reached its maximum only when the temperature was optimal. So we studied a lot of things about this graph. In this lesson, we will study another new keyword, limiting factor. They love it. And this is actually what they look for when they ask you about this graph relating to plants or photosynthesis. At A, temperature is the limiting factor. Okay? I hope you understand it. Now, another graph. This graph also, it is showing you that they are changing the light intensity and studying how the change in light intensity is affecting the rate of photosynthesis. Please pay attention to graphs. They love graphs. Okay. Again, we will divide this graph into A and B. At A, I want to describe and I want to explain. Fine. Now, can you describe A? What will you say at A?
As the light intensity increases, the rate of photosynthesis increases.
Excellent. Now, can you explain? The explain part is a little bit tough, but why, when the light increases, photosynthesis increases?
The particles gain more energy.
At A, the limiting factor is light intensity.
Yes, you are correct. At A, the limiting factor is light. If you want an explanation, because light, you will not be asked for an explanation when it comes to light because you didn't really take it, because light is the energy that will combine the carbon dioxide and water and convert it into glucose. This is part of the light-dependent reaction that takes place in thylakoids and chloroplasts. You will take it in A2 biology. So the explain part for O level, the only thing that you will be expected to say is that at A, the light was the limiting factor. How did I know? Because let's say when the light was 2%, the rate of photosynthesis was 5, whatever, okay, 5 arbitrary units. When the light intensity increased to 4%, the rate of photosynthesis increased also. So this means that at 2%, what was limiting the rate of photosynthesis was the light. So it is actually very easy. The part of the graph that is moving upwards, its limiting factor is whatever plotted on the x-axis. So at A, the limiting factor was light. What is limiting factor? This is the factor that will limit or stop photosynthesis from reaching its maximum. Now let's look at part B of the graph. If I want to describe part B of the graph, what will I say? I will start and help you. As light intensity is increasing, what is happening to the rate of photosynthesis? Is it increasing or is it constant?
Constant.
Constant. Okay, so at part B of the graph, you have to mention the word constant, or there are other words that have the same meaning. Either it will level off, you will see it in the mark scheme, or it will become constant, or it will reach a plateau. They like also this term, okay? In other words, at B, you will say that I'm increasing the light, increasing the light, increasing the light, but even though that I'm giving more light, the rate of photosynthesis is constant. So what is the limiting factor at B? If it is not the light, what can be the limiting factor at B?
Temperature.
Yes, you are correct. Any other factor? Carbon dioxide concentration, temperature, any other factor but not the light, because I am providing light. Even though I am providing light, the rate of photosynthesis is constant. Okay, good. Now, this graph, it's very similar to the one that we just talked about. But here, what is plotted on the x-axis? Another raw material, which is carbon dioxide. Now, again, this graph will be divided into A and B. At A, if I come to describe, how will I describe?
As the carbon dioxide concentration increases, the rate of reaction increases.
Very good. What is the limiting factor at A?
Carbon dioxide.
Correct. Perfect. Okay. So at A, the limiting factor was carbon dioxide. Why did I say that? Because I proved it. When I gave more carbon dioxide, the rate of photosynthesis increased. Now, how do you describe B?
As the concentration of carbon dioxide increase, the rate of the reaction is constant.
Excellent. Then what is the limiting factor at B?
Light or temperature.
Excellent. Any other factor? Anything other than CO2? Okay. I'm very happy that you're giving me correct answers. But does anyone have any questions? Anyone wants me to repeat anything? I hope it's clear. So I hope you understand the big picture. We will do an yes. Anyone asked?
Can you repeat what a limiting factor is?
Of course, my dear, I can. Look, my dear, we are studying photosynthesis. I'm going to explain the big picture from the very beginning. Photosynthesis is carbon dioxide and water in the presence of light and chlorophyll to give me glucose, C6H12O6, and oxygen. We will balance that now. We will ignore the glucose and put a coefficient of 6 before every other molecule. Okay, so photosynthesis means that in order for photosynthesis to be successful, I need carbon dioxide, I need water, I need light, I need chlorophyll, and I produce oxygen and I produce glucose, and this glucose will be converted immediately into starch. So I am going to do experiments and I will see if I change the carbon dioxide, what will happen to the rate of photosynthesis? If I will change the intensity of light, what will happen to the rate of photosynthesis? If I remove chlorophyll, what will happen to the rate of photosynthesis? I will do an experiment to prove that the gas that is produced is oxygen. This is the whole idea of all the experiments that we are doing. okay? We will change carbon dioxide concentration by either giving more CO2 or removing CO2. Let's look at light, because the light experiment is easy. I am going to set up this experiment in which I'm going to change the distance of the lamp. The closer the lamp, the more the light intensity, and the further away the lamp, the less the light intensity. Do you understand the experiment itself?
Yes.
Okay, very good. Whoever is asking, I hope the experiment is clear. And then I'm going to get some values like we always do, and then I'm going to plot a graph. My graph will look exactly like this. The temperature will be, where is my board? Yeah, here. I don't know, why does it go away? Okay. My temperature will be on the x-axis. Why? Because this is the value that I am changing. This is my independent variable. And my rate of photosynthesis will be on the y-axis, because these are my results. This is my dependent value. And I'm going to get a graph. It looks like this. The graph shows me that at a particular temperature, let's say at 40 degrees Celsius, the rate of photosynthesis was maximum. And I have two different parts on this graph. I have part A and I have part B. Let's look at B first. At B, it's very obvious that when I heat the temperature, when the temperature rises, the rate of photosynthesis will drop. And I think you know why. Because the enzymes will what? In the plant, the enzymes will?
Denature.
Denature. So this part, I hope it's clear. And we will elaborate on denature. Now at A shows me that every time I'm increasing the temperature, the rate of photosynthesis was increasing. If I will choose a particular temperature, let's say when the temperature was 5 degrees Celsius, the rate of photosynthesis was very low. Why? What was stopping the rate of photosynthesis from reaching the maximum level?
The low temperature.
Exactly. The temperature was very low. So the low temperature was the limiting factor that was stopping photosynthesis from reaching its maximum.
Okay.
Okay, my dear? So at A, anything on the x-axis, whether temperature or light, etc., as long as we have part of the graph that is going upwards, any factor or any independent variable on the x-axis was the limiting factor that was preventing it from reaching its maximum. And to prove that, when I increased the temperature to 40, the rate of the reaction now became at full potential. Clear? So a limiting factor, I'm very happy that it's clear. A limiting factor is any factor that will stop a process from reaching its maximum.
Okay.
Okay, very good. Now, you're welcome, my dear. Now here is a summary. By the way, we finished plant nutrition. Plant nutrition is very easy. We finished plant nutrition, and this is a summary here. I just want to point out certain things that should be understood, not memorized. Before any experiment that involves plants, we have to de-starch the plant before testing. I hope this is clear, especially if you attended last class. I have to remove the starch, because if I do not remove the starch, every time I will do a starch test, it will be positive. I want to first de-starch by putting it in a cupboard. I'll put my plant in a dark place for 2 to 3 days only, not more than that, because I do not want to kill the plant. I just want to remove the starch. And then I'm going to do my experiment. And after my experiment is finished, I'm going to test for the presence of starch. Now, either my results are positive or negative. If they're positive, then photosynthesis took place. If they're negative, then photosynthesis did not take place. Okay, I hope this point is clear. Now, I think now you know what is a limiting factor. Is a factor that will limit or stop a particular process from reaching its full potential. Okay? You don't have to know what the soda lime, or you don't need to memorize it, because they always give you this piece of information in the questions. Now, remind me, what was the test for oxygen? Remember the video that we saw? What was the test for oxygen?
If the gas is oxygen, it will relight a glowing splint.
Okay, perfect. Okay, now Photosynthesis again, respiration is the opposite. Please don't forget, photosynthesis requires light. So in any experiment, when you see a plant exposed to light, then this plant is producing oxygen because it is photosynthesizing. When the plant is put in the dark, then this plant will respire. And the plant in the morning, it will also respire. Respiration happens the whole time. Respiration never stops. But in the morning, the rate of photosynthesis is more than the rate of respiration. When we solve paper 2 questions, you will understand why I am stressing on that. Okay, we did talk about the structure of the leaf and the different layers of the leaf. Now we will discuss the second lesson, which is also really easy. It's just a continuation. The transport in plants. We will talk specifically about the xylem and the phloem. Now, the xylem and the phloem, we did study the xylem in the lesson, I think organization of the organism, because it was one of the specialized cells that we studied. Okay, I'm going to jump directly to a whiteboard. Give me a second. Because I'm going to draw the xylem. The best way to study the xylem is to draw it.
Miss,
Fine. Yes, my dear.
I wasn't able to attend the other sessions because it wouldn't let me because I was in another country and I feel like I missed out on a lot. So are
I
any of
sent you
them
the recording.
really?
Of course I did.
I didn't see it.
Why not? Because I sent the recording immediately on the group for everyone. After 1 hour, I sent the recording and I put it on Google Classroom.
Okay.
Okay.
Thank you.
So what I'm going to draw now, I'm going to draw the xylem. What is the xylem? To cut a long story short, the xylem is a tissue. It's not an organ. It's a tissue that is found in plants. And this xylem transports water, you should know, and ions and mineral ions and salts. Now let me draw the xylem. When I come to draw the xylem, I'm going to draw something that will take me exactly 30 seconds to draw, because this is what I'm going to draw. That's it. I will not draw anything else. I can add something so that I don't forget. Thick walls. Now, number 1, where is the xylem? The location of the xylem. The xylem is in the center of the stem. If this is the stem of the plant, the xylem is in the center of the stem. This xylem carries water and mineral ions. Okay, I'm going to look closely at the structure of the xylem. I just drew it, and I simply drew an empty tube, because the xylem is an empty tube. There is nothing inside the xylem. Why does it disappear? Okay, now the xylem, each one of those is a cell. Do you see any nucleus inside the xylem cell? Do you see anything inside it actually?
No.
No. The xylem is a specialized cell. The xylem is a specialized cell that carries water. This xylem, if you look closely inside it, you will find that it has no nucleus. This is really important. No cytoplasm. No organelles. You will tell me it doesn't have anything? Yes, it is completely empty, and it is even considered dead. Now, why is it without a nucleus and without a cytoplasm and without organelles? Why is this empty?
Make it hollow.
Exactly. Perfect. Because the xylem cells, as you can see, they are arranged on top of each other to create a hollow tube. What you said is perfect. We want to create a hollow tube, because this hollow tube will carry water, and we want the water to move upwards with no obstruction. Not only are these cells completely empty, but their end walls, see, this part is even broken down. They have broken down end walls. So they love the xylem, and they love you describing the xylem. So how do you describe the xylem? You will simply start by saying it is a tissue that transports water and ions. It has another function also. It provides support, and we will explain why. This is how you will draw the xylem. That's it. It will take you 30 seconds to draw the xylem, and you can also draw arrows moving in one direction only, from the soil to the leaves, from bottom to top. These arrows represent the movement of water and mineral ions. Now, describing the xylem, let me change color. Describing the xylem, don't forget, it's a tissue. What is a tissue? A tissue is made up of similar cells that work together to carry out a particular function. It is made up of cells, many cells that will create the xylem. Okay, so the xylem is a tissue. It is part of the transport tissue. Because what is the function of the xylem? Actually, the xylem has two functions. Function number one, conduction. Conduction means it carries water and ions from the soil to all parts of the plant. The second function of the xylem, it gives you support. The xylem, the cell wall of the xylem is not like the traditional cell wall in plants. The cell wall of the xylem, not only does it contain cellulose, but it contains cellulose plus another molecule known as, who knows it? Known as lignin. And this lignin is very, very strong. If you want to know how lignin, how strong it is, you know the tree and you know the bark of the tree, this brown bark of the tree. This is lignin. Are you all with me? So we are still talking about the xylem. The xylem, two functions. First, it's a tissue. It's not a cell, it's not an organ. It's a tissue. It's made up of similar cells that carry out a particular function. Now, what is that particular function? The xylem has two functions. One, it carries water and ions, and this function is called conduction. And the second function, because it has very thick cell walls, very strong cell walls, the second function is that it provides support. Is anything hard in what I said? Any questions? Hello? Anyone has any questions?
No, thank you.
Okay, I didn't finish. Now, the structure of the xylem, we finished the function. The structure, to remember the structure, you draw it. I'm going to draw empty cells on top of each other with very thick walls and a broken down end plate. The edges are broken down. They create a hollow tube, just like your friend said. Now, if I come to describe the xylem, what do I say? I'll start with the word no. No what?
Nucleus.
No nucleus.
Cytoplasm.
Also no what? No?
Cytoplasm.
No cytoplasm. No what?
Organelles.
Very good. Very good. No organelles. It is considered
Dead.
dead. Very good. And what about the end walls? These end walls, are they completely or incompletely broken down?
They're completely
broken down.
Completely broken down. Very good. If I write all that, then I will describe the xylem, completely broken down. Now, why are the end walls completely broken down? Because what do I want to create? What do I want to create?
hollow tube.
Very good. I want this keyword. I want to create a hollow
Hollow
tube
tube.
that will carry water, and this water will move smoothly without any obstruction. And this water always moves in one direction only. This is also important to know, that the water in the xylem moves only in one direction, from down to up, from the soil to the leaves. Also, the cell wall of the xylem, if I
How
want
does the
to...
water move up, Yams?
Excuse me? How does the water move from where?
How does update Xylem?
I'm going to answer this question because it's going to come next, but I want to ask you something. Where does the water come from anyway? What is the source of the water? Are you all with me? I think we lost connection. You're all with me now?
Yes.
Yes.
Okay. Now I was answering a question. How does the water move? We will discuss this point because this is in your lesson. Something else. The water comes from where? The water comes from the soil or from the leaves? When you come to give the plant water, where do you spray this water? Where do you add this water? Who was asking?
By the roots.
Yes, from the soil, from the roots. So the water, okay, the water, where is the concentration of water higher? In the soil or in the leaves? I'm asking you, whoever asked me, water, where is the water more concentrated? At the base of the plant or at the top of the plant? In the soil. So actually, I
Base.
have lots of water at the soil. I have lots of water at the base of the plant. Water always moves by osmosis from high to low concentration. This is always the direction of water. Water will always move from the high concentration, which is the soil, to the leaves of the plant. Okay? Now, and I'm going to
Yes.
answer this question in more detail when we move on with the lesson. Okay, going back to transport in plants. We will study the xylem. Going back to the two tissues that are found in the transport tissues of the plant, xylem and the phloem. We have the xylem and we have the phloem. Now we did study the two functions of the xylem, 1 and 2. What's the first function? Conduction. Conduction simply means the transport of water and not the transport of water only, and mineral ions, because these ions, the magnesium and the sodium and the potassium, they are all dissolved in the water. Okay? Number 2, support. And we did elaborate here. We said that the xylem provides the support because the xylem walls are very strong. The walls of the xylem, they contain cellulose like any other plant cell, plus what else? Cellulose plus, who remembers what I said? Lignin. Can you say lignin? Lignin is a carbohydrate that is very strong, that provides xylem with a lot of support. Now there's another tissue that is known as the phloem tissue. This phloem tissue, it does not carry water and ions. It will carry something else. What does it carry? Who knows? We took it in year 9 also. What does the phloem carry? Wake up with me. The phloem carries what?
Miss, your voice is lagging.
My voice is lagging? Really?
Yes.
Oh,
Yeah,
okay.
it's very.
There's a problem with the okay, then I will have to repeat. I was asking about the function of the phloem. What does the phloem carry? Is my voice clear now?
Yes.
Okay. The phloem. We said that the
Sugars, amino acids.
yes, sucrose and amino acids. Okay. If you're confused, don't worry. We will elaborate. Now, this is your syllabus. We'll go to the source and the sink because this is whatever. Now, let's go back to the different layers of the leaf because I want to know the exact location of the xylem and the phloem. This is the waxy cuticle. This is the upper epidermis. I hope you already know this by now. This is the middle layer or the mesophyll tissue that is divided into the palisade mesophyll and the spongy mesophyll. These contain chloroplasts and they do photosynthesis, and these are the air spaces for the exchange of gases and the circulation of gases. And here we have, these openings are called what? stomata, and they are controlled by guard cells. Now, this is the transport tissue. This is called the vascular bundle or the transport tissue, any one of those, so that you are familiar with both. And it is made up of xylem or phloem. Each one of them transports something different. The xylem transports water and ions dissolved in the water, and the transport of water and ions is called conduction. And the phloem does not transport water. The phloem transports food, the plant's food, which include sucrose and what? Amino acids. Very good. And the transport of food, the transport of sucrose and amino acids, it's not called conduction. It has another name. Who remembers the name? It's called, starts with a T also.
Translocation.
Translocation. Okay, I don't know what happened to my pen. Translocation. Good. Now, the location of these vascular bundles in different parts of the plant. We will take part of the stem. I'm going to take part of the stem, take a cross section of the stem, and I will look inside. And I am expected to know the location of the xylem and the phloem. Now, there's nothing to understand here. You just need to memorize the location of the xylem and the phloem. If I look at the stem from above, or this is a cross section of the stem, I'll find that the xylem is always located towards the inside of the stem. And the phloem is always located towards the outside. You will receive lots of questions in paper 2 asking you about the location. So if I take a section of the stem, I will see that the vascular bundle is located in a ring arrangement, and this ring arrangement provides support. And it is divided into 2 sections, xylem and phloem. The xylem is always towards the center, so this is the xylem, and the phloem is always located towards the outside. Now let's look at other pictures. This is a cross-section of the root. Now, why do I know that this is the root? Because I can see that the vascular bundle is in the center. And this is my root, and the vascular bundle is in the center. The vascular bundle is the bundle that contains both the xylem and the phloem. Where is the xylem and the phloem exactly? It looks like a flower, just like you see in the picture, and the xylem is in the center of the root. And the phloem surrounds it. So this blue part is the xylem, and this yellow part is the phloem. Let's look at other pictures. This is a picture of the xylem and the phloem, the pictures that I was drawing, trying to explain to you the differences between the xylem and the phloem. We only drew the xylem. We did not draw the phloem yet. The xylem is actually what is requested from you. You are not requested to know the structure of the phloem. You are only requested to know the function of the phloem. But the pictures are there so that you can compare between them. As you can see, the xylem is made up of cells with completely broken down end walls. These end walls are broken down and they are empty, no water, no nucleus, no cytoplasm, no organelles, so that the water can move smoothly up the xylem. Let's read the labels. Here, what is transported? Water and minerals only. One-way flow of sap. You don't have to know the word sap for now, but the direction is one way, always from the soil to the leaves, always. Thick cell wall made up of what? Cellulose and lignin. Lignin is only found in the cell wall of the xylem. This is supplement material. This is important. And that is why the walls of the xylem are very strong. If you want to imagine how strong they are as strong as the barks of a tree. You know the bark of a tree? This wooden material is lignin. So I hope you can imagine how strong lignin is. And this gives an additional function to the xylem, which is support. The xylem helps in supporting the plant. Cells have no end walls. See, these end walls are completely broken down between them. Now, why don't they have any end walls?
Because it's hollow. To create a
Because we
hollow
want it to be hollow, correct? Because we want the water to move up. Now, let's compare the structure of the xylem with the structure of the phloem. You will not see any questions over the structure of the phloem. But why is it there in your notes and why am I explaining it? Because they will give you a diagram like this exactly, but without the labels. And they will tell you, compare. They will tell you which is the xylem and which is the phloem. So you have to know the difference in terms of structure. You have to know the location and you have to know the different functions. Okay? The xylem is very easy to identify because it has very thick walls. See, it is even drawn in brown because it makes up the barks of the tree, and the end walls are completely broken down and it's empty. It is considered dead. Now, the phloem is a different story. The phloem, the cell walls or the walls of the phloem are much thinner. The phloem, it has no nucleus, yes, but it does have a cytoplasm. It has a cytoplasm and it does have a few organelles. The phloem is not considered dead. The phloem is considered living. And the end walls of the phloem, as you can see, they are not completely broken down like in the xylem. They are partially broken down. They create something that looks like a sieve. Now, to explain what is a sieve, I will just cut a long story short and I'll say it in Arabic, even though I don't like that. You know the masfa? You know the sieve that you use to separate like foods from each other? Masfa. You know the masfa? You know it? You know what I'm talking about? You know the sieve?
Yes.
The one that you have in the kitchen? Very good. So the end walls of the phloem, they look like a sieve. And they are described in biology as the sieve tube element. You don't have to know the structure of the phloem, and I will say this 10 times. You don't have to memorize the structure of the phloem. You have 2 tables in the notes. You will study the xylem. You will look at the phloem just to compare, but you don't have to study the structure of the phloem. But you have to know that the structure is very different. It does contain a cytoplasm. The phloem does contain a few organelles. It is considered living. The wall of the phloem is much thinner. And the end walls are not completely broken down like the xylem, but it is partially broken down to form something that looks like a sieve. And they actually call it the sieve tube element. And it transports sucrose and amino acids. And it is incorrect to say that it transports carbohydrates. They want sucrose and amino acids. And look at the arrows. The sucrose and the amino acids, they move in one direction or in different directions.
Different directions.
Different directions. So if you are actually asked to compare between the xylem and the phloem, you will have a lot of things to say. Okay, you will use the table that you have in your notes. You can only study the xylem and the phloem is the opposite. The xylem is dead. The phloem is not dead. The xylem has no nucleus. The phloem also has no nucleus. The xylem has no cytoplasm, but the phloem, it does have a cytoplasm. The xylem has no organelles at all. The phloem has a few organelles. The xylem carries water and mineral ions. The phloem carries sucrose and amino acids. The xylem carries the water in one direction only, but the phloem, no, it carries the sucrose and the amino acids in all directions. The xylem has very thick walls and it is very strong. The phloem has thinner walls and it does not provide support. The end walls of the xylem are completely broken down. The phloem, the end walls are not completely broken down. They're partially broken down. They form something called the sieve. Okay, so here I have sucrose and amino acids, 2-way direction, thin cell walls made only of cellulose, no lignin in the phloem. And these cells with end walls and perforations. These are the perforations because they are incompletely broken down, creating something that looks like a sieve. Okay, and this is the table that I'm telling you about that will compare between the xylem and the phloem. Let's quickly look at it because they love the comparisons. Xylem transports water and minerals. It is strong. It provides support. I hope number 1 is clear. Phloem, it carries food, sucrose, and amino acids. The transport of water is called conduction. Please stress on the keywords. And the transport of sucrose and amino acids is called translocation. Xylem, made up of dead cells, which are joined end to end with no end walls, no cross walls, to form a long, continuous hollow tube. Okay? These end walls are completely broken down, forming a hollow tube. This is very important. You'll see it many times in the mark scheme. The phloem, no, it's different, made up of living cells. The end walls are not completely broken down, but they contain holes or perforations or sieves to form sieve plates, and they allow easy flow of substances, sucrose and amino acids, not water. And the cells are called sieve tube elements. I hope everything is clear. If you have any questions, stop me. Again, I'm comparing between the xylem and the phloem. Xylem, no cytoplasm, no nuclei, no organelles. It's hollow, it's empty, it's dead. Okay? And this is important because it will make the xylem adapted to its function, which is the carrying of water without any obstruction. The xylem vessels act as a capillary tube. You will see also this word in your mark scheme. Please, when you study and you see a word that is in bold, give it some attention because this bold word is a keyword that helps to pull the water upward. We will explain the pulling of water part in the next lesson. The phloem contains cytoplasm but no nuclei. But it does contain cytoplasm, the phloem. That's why it is considered living. More differences. The cell wall of the xylem or the walls are thick, made up of cellulose and lignin. Phloem, their walls are thinner, made up of cellulose only, but no lignin. Are you all with me? Can you all hear me clearly, or is my voice lagging?
Yes, it's clear.
It's clear.
Okay. Now let's skip number 5. I'll go back to it later. 6. Water in the xylem move only in one direction, upwards. This is because transpiration takes place from the upper part of the plant. Also, let's ignore this point. I'll explain it in detail when we discuss transpiration. Phloem, the sucrose and the amino acids moves in all directions, not only in one direction, in all directions. Okay? Now I will mention one last thing and then promise I will let you go. Okay? Now this picture is very nice. This is a real picture using an electron microscope of the sieve or the end walls of the phloem. This is what I was trying to explain, that the end walls in the phloem, they're not completely broken down like in the xylem. They're partially broken down to give you sieve-like cells. They look exactly like this. This is a sieve tube element. See? There are holes here. This is incomplete breakdown of the end walls of the phloem. I hope it's clear. Fine. This is a scanning electron micrograph of a sieve plate in a phloem tube. Good. Now, the last thing I wanted to mention, again, this is a picture of the phloem tissue. As you can see, you have cells on top of each other, just like the xylem, but here I have incomplete broken down end walls that look like a sieve plate, so they called it the sieve tube element. Now, adjacent to every sieve tube element, there is a cell known as the companion cell. Now, you will not need to know anything about this companion cell in O level. We will study it later in year 11. The only thing that you need to know about the companion cell is that you have one cell adjacent to every phloem cell. It's like a companion or companion. That's why they called it a companion cell. And it has one function for now. You only need to know one function, that it will control what will move in and out of the phloem. So again, let's compare between the xylem and the phloem. The xylem, I want to study the xylem and the phloem. I will draw it in 1 second. That will hopefully remind me of the differences. For the xylem, I will draw empty cells on top of each other. That's it. with thick cell walls. This is all what I will draw for the xylem. I can add an arrow, and this arrow will be in only one direction, from bottom to top. This will help me describe the xylem. No nuclei, no cytoplasm, no organelles, dead, hollow tube, very thick walls made up of cellulose and lignin. The end walls are completely broken down, and this will help in the unobstructed flow of water from down to up from the soil to the leaves. I finished discussing the xylem. Two functions: conduction, transport of water, and support, because it has very thick walls, so it provides support. I am done with the xylem. Any questions about the xylem? Any questions? Can someone say yes or no?
No.
No.
Okay, stay with me. The phloem. I want to study the phloem. I will draw it. Always start by drawing. Drawing really helps you. I am going to draw also cells on top of each other, just like the xylem, but I'm going to make a few additions. Number one, the walls are thin, they're not thick. Number two, the end walls are not completely broken down like the xylem, but they are perforated, like this, creating something that looks like a sieve, okay? And I'm going to draw arrows in the xylem, these in the phloem. These arrows, they move in one direction or in all directions?
In all directions.
Very good. I'll make an arrow going up and an arrow going down. By the way, they do accept small diagrams in the answer. You can make a small diagram as long as it is within the space provided for answering. So this is the phloem. This will hopefully remind you that the walls are thin, so it does not provide support like the xylem, and the movement of substances is up and down. And no nucleus, yes, but I do have a cytoplasm. I do have a few organelles. So here it is considered living. It is not considered dead, but the xylem is dead. Don't forget that, okay? And here, the transport of sucrose and amino acids is called translocation. And adjacent to every phloem cell, there is a companion cell that looks like this. This cell is like any other plant cell that has all the organelles and the cell walls and all the structures of the plant cell. And it has only one function. It will control what will move in and out of the phloem. You don't have to know anything about it, other than there is a companion cell next to every sieve tube element in the phloem that will control what moves in and out of the phloem. This is the companion cell. So just by drawing, I will compare the xylem and the phloem. Anyone has any questions? Hello? Anyone has any questions?
No.
No.
I'll go back to the table because I wanted to go back to number 5 that we said we will talk about it later. In the xylem, no companion cells, but in the phloem, each sieve tube element means each phloem cell. It's another name for a phloem cell, has a companion cell next to it. So I hope now number 5 is clear. That's it. You just need to know that there is a companion cell in phloem and no companion cell in xylem. Any questions?
No.
Any questions? Okay, I will revise what we took today. This lesson is really very easy, but the questions are very frequent, so I'm just stressing on it. Today we discussed the graphs, and these graphs are very easy to understand. What is new that we took today is the limiting factor. The limiting factor is a factor that will limit photosynthesis, that will stop photosynthesis from reaching its full potential. The limiting factor is any part of the graph that is moving up. What is the limiting factor? What is stopping photosynthesis from reaching its maximum potential?
Temperature.
Okay, and at B here is the optimum temperature. At C, the enzymes denatured. I think you know this very well. Also, This graph with the light intensity on the x-axis, what is the limiting factor at A?
Light intensity.
Very good. At B, what is the word that we use here? The rate of photosynthesis is?
Constant.
Excellent. It's constant. What is the limiting factor at B? Is it light?
Any other factor except light.
Thank you so much. Thank you so much. Carbon dioxide, temperature, etc. And the same here. At A, carbon dioxide is the limiting factor. And at B, any other factor is limiting, whether temperature or light or whatever. Okay, I hope the graphs are clear. And then we talked about the xylem and the phloem in detail. We studied their function, their structure, and their location. I hope this is clear. I will not go over the structure and the function again. I think I made you very bored today, but I will go over the location quickly and hopefully I'll let you go. I'm going to talk about the location of the xylem and the phloem in the stem and in the root. I'm going to take a cross section and look at the vascular bundle in the root. Where do I find it? Arranged in a ring or in the center? When I'm talking about the root, the vascular bundle will be in a ring in the center. Thank you very much. It will be in the center. And the xylem and the phloem will look like a flower. I'm going to change color. The yellow one is the xylem or the phloem? The yellow xylem. Thank you very much. So the yellow part is the xylem. And the phloem is the tissue in. Very good. Thank you. Now in the stem, I'll find the vascular bundle arranged in a ring. Like this. It looks like a triangle like this. And each bundle is divided into two parts. The part that is directed inwards is the xylem or the phloem?
Xylem.
Xylem. Very good. And the outer part is the phloem. Nice. Now, promise I'll let you go one day. I have the leaf. I'm looking inside the leaf. Again, I have the vascular bundle here in the mesophyll layer divided into two parts. The upper part, the part facing towards the upper surface of the leaf or the upper epidermis is the
Xylem.
Thank you very much. And the lower part is the phloem. There's nothing to understand here. You just need to memorize their locations. That's it. Where is the picture? And then I'll let you go. Promise, one day I'll let you go. Here, this is the root. Because the vascular bundle is in the center, the xylem is in the center of the center, and the phloem is on the outside. This is my leaf. The xylem is up there towards the upper epidermis, and the phloem is at the base of the leaf. This is the stem. They are arranged in a ring arrangement. The xylem is always towards the inside, and the phloem is always towards the outside. You have all these pictures in your notes. So, okay, nice pictures here. The root in the center. Don't forget, the vascular bundle is in the center. The stem, they are arranged in a ring. Xylem towards the inside, phloem towards the outside. Okay, I promised I'll let you go, so I will let you go. We have 2 points missing to finish this lesson, which is transpiration. We will discuss transpiration in detail and the factors or the methods by which plants that live in dry environments prevent transpiration, and we already took it in adaptation, and the source and sink. This is also supplement material that we will take together. Okay, any questions before I go?
No.
No.
Okay, if you have any questions, send me. Fine. Some of you are lovely and amazing, and they finished the homework, which was paper 2 and 4 drugs. And I keep on telling you, what else do I do? You know what? I'm going to give you an assignment. This assignment will be paper 2, 4, and 6 plants. This is a very long assignment, so I'll give you 2 weeks. Okay? And we will start working on it together. Don't worry. We will solve a lot of it together in class. Fine. So I'm going to create a new assignment to solve paper 2, 4, and 6. Ignore paper 4 for now because we did not finish the lesson. And this assignment will be for 2 lessons. Lesson 5, plant nutrition. And lesson 7, which is transport in plants. Because we always take these 2 lessons together. We finished plant nutrition. Plant nutrition is very easy. We only have transport and plants missing. Okay? If you have any questions, send me anytime on WhatsApp. Bye-bye.